Okay, good morning and welcome to our third scientific seminar of the year. After all these months without scientific seminar, we are today recovering this activity in remote mode. And I would like to thank the organizers behind this seminar, in particular Maria, that is here with us for the work that has been done to really prepare for this. Maria will be also dealing with the questions and answer part of the seminar. Okay. Seminar in particular, Maria, that here with us for the work that has been done to this. Maria will be also dealing with the questions and answer part of the seminar. So, okay, is that okay? Okay. So as today, our speaker is Professor Enrico Coen. As I was saying, is known to many of us because he has been working with us in some panel in the evaluations of the ERC calls. But before starting with his seminar, let me just share with you just some elements of his biography. Enrico was born in Liverpool in 1957 and obtained a PhD in molecular genetics in Cambridge University in 1982. After a postdoc at Cambridge, he moved to the John Innes Center in Norwich in 1984, where he began using anti-rhinum as a model system to study plant development and evolution. Based on the results of an extensive mutational screen, he proposed that the ground plan of a flower depends on the combinatorial action of homeotic genes acting along the radial and dorsoventral axis of the flower. More recently, he has started to explore ways of bridging the gap between gene actions and the development and evolution. It has involved collaborations with computer scientists and population geneticists to develop methods for quantitative modeling of plant growth, form, and diversity, a truly interdisciplinary research. In addition to publishing in scientific journals, Enrico Coen has also tried to communicate some key principles in the area of a broad audience through articles in popular journals and his books, two very interesting books that he wrote, The Art of Genes and Cells to Civilizations. In these books, he explores the connections between science and art. He's also a fellow of the Royal Society and a foreign member of the US National Academy of Sciences. He has also had an LS3 advanced grant, and as I said, he really collaborated with us in 5 occasions, being a panel member for LS3. The title of his talk today is From Planes to Caps, the development and evolution of leaf shape. Today's seminar will be quite special in the sense that Professor Coen has been kind enough to pre-record his presentation. Okay, we will have the added value that we will have subtitles in it. Okay, so I hope we will all enjoy the presentation. And without further ado, we will go now through it. watching it, and Professor Coen will join us for the Q&A part of the seminar after we finish watching the presentation.
Hello. First of all, I'd like to thank the ERC for inviting me to give this talk. I was due to give it in March of this year, but because of COVID, that wasn't possible. Nevertheless, I'm looking forward to sharing our research with you in this way, and very much look forward to hearing the questions you may have at the end. I will also try and flag how ERC funding has really made a big difference to this research project. What I'm going to be talking about is a fundamental problem in biology, which is how the remarkable forms and shapes and forms that we see around us in biology are generated through development.
Each of these shapes, the daffodil, the brain, the leaf, the butterfly, the pitcher leaf, and the heart, all arose through sheets of cells somehow miraculously deforming themselves into these different forms. And the question I want to ask is, how did that happen? And might there be some common fundamental rules by which such transformations occur? Now, the idea that there might be unifying principles or rules behind a whole series of different transformations isn't new. A few years ago, I attended a conference in which the poster for this conference showed a series of creatures or forms generated through origami. Now, origami involves a series of basic transformations or basic folds, and it's the combination and the way you make these folds that generates all these different forms. But the fundamental principles are actually very simple, just by folding a piece of paper. The question is, might there be an equivalent set of rules or principles that underlie all the varied forms that we see in biology? Now, unlike origami, however, in biology, we don't have an external hand that's doing all the folding, that's doing all the deformations or transformations that we're seeing here. It's all occurring through the actions of genes within tissues. It's internally driven through the properties and behaviors of the cells in a tissue. So the question is, what might be the principles through which the equivalent of origami occurs in biology? And to illustrate that, what I'm going to do is try to do a kind of biological origami, the equivalent of origami, and see if we can achieve it, but through biological properties, internally driven, rather than through externally imposed hands performing the operations. So our starting point for this bioorigami, as I'm going to call it, is going to be a sphere of tissue. You can think of it as a sphere of cells collected together. And this tissue, the first problem we're going to have is to generate our sheet, because origami starts with a sheet, but in bioorigami, we're going to have to somehow generate that sheet to begin with. So how might we do that? Well, to generate a sheet, we need some sort of orientational information that's going to allow us to sort of grow or deform this sphere in one direction more than another. And to do that, I'm going to introduce the notion of what I'm going to call a mid-plane identity, an equatorial region that runs through our entire sphere that is going to help us establish this orientation system. Now, just to show you, this region in green, which I call the mid-plane identity, runs through the entire sphere. I'm going to cut a section through it so you can see it runs all the way through. And you can think of this as a region of gene activity that is somehow defining an equatorial plane or mid-plane. Now, in order for such a thing to help us orient growth, we're going to need to have, as I said, some sort of orientational information. And so to do that, imagine there's something propagating out from this midplane. It's producing something that, say, diffuses away from the midplane, and that we're able to take the gradient of that substance. So if you can take the gradient of this substance or propagating material that comes out of the midplane, we'll get a set of arrows. So the arrows tell you the steepest gradient of propagation. All right, so the steepest gradient of decline in the concentration of this substance. Now, once we have a set of arrows like this, which we call a polarity field, we can define two types of growth. One type of growth is we can call parallel to the polarity field, K-par. So that's growth oriented parallel to the local polarity. So for each region of tissue which has an arrow, we can say the tissue might grow at a certain rate parallel to the orientation of that arrow. And we can also define a second growth, which we can call perpendicular to the polarity. So it's in the plane perpendicular to that polarity. So growth can also be oriented like that. And so we can play with these two different growth rates for each region of our tissue. We can say to what extent does it grow parallel or perpendicular to the polarity. So in my first illustration, what I'm going to say is suppose now growth is only perpendicular to the polarity. So that in each region, growth is going to be high, perpendicular to the polarity, and little or absent in terms of parallel to the polarity. So K-per is uniformly going to be high throughout our tissue. So if we grow our tissue in that way, what will happen to our sphere? Okay, so if we do that, what happens is the sphere flattens. It grows into a flat sheet. And just to complete the tissue, we end up with a disc. So our sphere has grown into a flattened disc by invoking this polarity it created through this midplane. Okay, now we've created a disc, a sheet. What if we want to bend the sheet, as in origami? So how might we do that? Well, here's an idea. Suppose that we have a strip of higher growth rate on the upper surface of our sheet. All right, so high growth perpendicular to the polarity on the upper surface of our sheet. Might that cause a bend? And the reasoning being that if we have more growth along this line and the top compared to the bottom of surface, that might somehow create a bend. So is that what happens? Well, if we run this, what you generate is a sort of bend, but it's not an origami fold. All right, we're not getting a nice, neat origami fold. We're getting a sort of curved fold. And so why is this? Why aren't we generating a nice, neat origami fold? And what would we need to do to the system in order to enable us to generate a nice, neat origami fold? Well, to answer these questions, I'm going to now give you a demonstration. Okay, so here I've got a piece of paper. It's a normal piece of paper from a printer. And the only strange thing about it is that I put tape on one side of the paper. And there are 3 strips of tape, one going along the top, one going down the side, and one diagonal strip. And what I'm going to do is just cut the top strip off to begin with. Okay, so I'm just going to do that. With these scissors. So here is the top strip. Now I'm going to take some water over here, and I'm going to put this strip into the water, and I'd like you to think about what's going to happen. Okay, so try and predict what's going to happen. So I put the strip into the water. Okay, and let's see what happens. You can see it starting to curl to form a ring. Okay, so why does it do that? The reason is that the paper, when it absorbs the water, wants to expand, okay, get bigger. But the tape doesn't want to expand. It wants to stay the same size, as it were. And so there's kind of a conflict between the paper growing larger and the tape remaining the same size. And by bending, the conflict is resolved because it allows the paper to get long while it allows the tape to stay the same size. Okay. So what's going to happen now when I take the vertical strip, the other strip? Will it do the same or something different? Okay, so let's cut that off now. All right, cut the ends off. I put
put it in the water. What do you think is going to happen? Take it out. Okay. So what's happening? It's curling again, but now it's curling in the other orientation. It's forming a long tube instead of curling on itself in the longitudinal direction. So why is that? What that tells you is that the paper is anisotropic. It preferentially expands in one orientation compared to the other, and that's why we're getting these two different results. The fibers actually in the paper are aligned in a particular way in such so that you end up with the paper stretching in different orientations. So now we've done that, let's take the final demonstration, the diagonal strip. And I'm hoping that you can predict what's going to happen. So now let's take that. I'll cut this off. Try and think what's going to happen now when we put the diagonal strip. In the water. Okay. Now I don't know how many of you anticipated this, but we're now getting a nice helix. Okay. So what this tells you is that the paper is anisotropic, and by having anisotropy or preferential orientation of fibers in the paper and preferential orientation of strength in the paper, we're able to orient the direction in which this folds on itself. Okay. So armed with that knowledge, we can now go back to our example of why the disc folded in a particular way. I hope you can appreciate now that the reason that it's folding in this strange way is that the growth in the upper surface in this line is now going in both directions. It's going both parallel to the line, creating this arch, and perpendicular to this orange line, creating the fold. And the result is we end up with it arching or bending in both directions. So if we want to get around this problem, what we need to do is somehow orient the growth in the plane of our tissue, in the plane of our disc. And one way of doing that is to create a second polarity field. So here now I'm showing you two polarity fields. One that you've seen already with the black arrows, what I call the orthoplanar field, that emanates from this midplane. The other one almost goes orthogonally or perpendicular to the blue arrow, to the black arrows, these blue arrows, and they go from one end of the equator to the other end. All right, so to creating an alternative, what we'll call a planar polarity field. And the reason that this is called a planar polarity field will become evident when we now try and grow our disc. So let's grow the disc just as we did before, orienting the growth with this orthoplanar field such that growth is perpendicular to it. All right, if we do that, but now show what happens to the planar polarity field, what we see as we grow it is that the blue arrows now get deformed and start to align in the plane of the tissue. All right, so I can show you that in a completed disc. So now we have a field of arrows, a planar polarity in the plane of our sheet. And now when we want to make our fold, we can specify three types of growth direction. There's what we call K-par, growth parallel to the black arrows. K-par2, growth rate perpendicular to this but parallel to the blue arrows, and a third growth rate, K-per, which is perpendicular to both. And if we have it such that the growth now in our blue line, is only going to be, which is on the top surface, only happens parallel to the blue arrows, parallel K-par2, in other words, it's only parallel to these blue arrows, what's going to happen? Let's have a look. It folds, it gives you a nice origami fold. So, to summarize, we can perform a kind of bio-origami, but to do so we need a number of ingredients. First, we need a set of regional identities, like that green midplane region, equatorial region. Then we need two orthogonal polarity fields. These are the blue and the black arrows, two sets of arrows that are going to allow us to orient our growth according to three different directions, as we've seen with this cross shown here. And finally, we need mechanics. We need the tissue to be mechanically connected together so that it forms in a coherent whole. And with these principles, we can do bioorigami. We can create these types of folds, but now using the internal rules of growth of the tissue. But we can also do a lot more. You may notice that with origami, we can only create a rather set of angular flat shapes. It's rather limited in what it can do. It turns out with these rules, we can do much more than origami. We can create rounded forms, forms that are much more typical of the things that you see in nature. So far, I've given you some illustrations of the principles by which tissues might be produced and deformed. But the question is this true? Is this the way that actually tissues do behave? And to evaluate that, we need to look at specific biological cases and see the extent to which what I've been telling you helps us understand what is going on. And the first example I'm going to give concerns how a flat leaf develops. This is a leaf of the workhorse of plant genetics, Arabidopsis thaliana. And we know quite a lot about the genes controlling the formation of this sheet of cells that forms the leaf. And to give you an idea of where these genes are active in the leaf, what I'm going to show you is a kind of a section, a small section through the leaf, looking in cross-section to see where these genes are switched on. And when you do that, what you see, this is shown on the left here, what you see is that we essentially find three domains of gene activity, three regions that are distinct. There's the light blue area, the top half of the leaf, which is called the adaxial domain. Then there's a sort of brownish colored area, which is the abaxial domain. And then finally, I've color coded the mid-plane region in green that runs between the two. Okay. Now, these genes don't just simply decorate or identify different domains of the leaf. They also are important in the formation of this sheet-like structure. Because if you were to mutate these genes, inactivate one of them, let's say, you end up with a leaf such as the one shown here, which is now radially symmetrical. It's a radially symmetrical mutant because we've lost the blue domain, we've lost the midplane. All we're left with is the abaxial domain, the brown domain, and the result is a radially symmetrical mutant. So to illustrate that, I'm going to show you it in a different species, Antirrhinum or snapdragon. So here is the normal snapdragon leaf, and here is one of these radially symmetrical mutants where these domains have been inactivated. So now I'm going to play that movie. So you can see that this radially symmetrical mutant really has lost its sheet formation, its inability to form a sheet. So how do we explain that in terms of the principles that I've gave you earlier? So let's go back to our sphere with its different polarities. If you remember the blue and the black arrows, and this third one, the orange one, which is perpendicular to both. And let's grow it again, as shown here. And remember that the reason it forms a flat structure is because growth is being oriented by the black arrows. That's what's keeping it. So growth is being oriented in the plane perpendicular to the black arrows, which is giving you this sheet-like structure. It's no longer a disc. It's narrowed at one end more to be like a leaf, because in addition to growth being oriented by the black arrows, we're also reducing growth parallel to the orange arrows at one end of our structure, which is what gives you this narrowing at one end. All right, so but using this, we can generate a leaf-like shape. And if we look what happens to the blue arrows, we again see they're in the plane of our sheet. Now, what would happen if we would take away the midplane? Use exactly the same model, but take away the midplane and lose those black arrows. Okay, well now we've lost the black arrows on the right, and we've now only got the blue arrows to orient our growth. All right, and if we now, because we've lost, in other words, our ability to make a sheet. So if we now run this model, exactly the same as before, but without the black arrows, we end up with a cylindrical tube. So we can explain the formation of our radially symmetrical mutant as a mutation that essentially gets rid of the black arrows of the orthoplanar polarity field. And so we only end up with one polarity field, and that's why we end up with this cylindrical form rather than the sheet that we see normally. All right, so we can understand these mutants and how they act in terms of growth being oriented by these polarity fields that I've been describing. That tells you something about how the sheet is formed, but what about the shaping of the sheet? Why does the sheet get shaped to produce a leaf with its characteristic outline? To understand that, we need to go and think more about how growth is oriented in the plane of the sheet, okay, after the sheet has formed. And we can do that by imaging the development of a leaf by fluorescently marking its cells. So here, for example, is a leaf bud of Arabidopsis, labeled with its cell outlines, labeled with a fluorescent marker. And if we grow that leaf, as it grows, we can track and follow all the cells, all the vertices that connect the cells, and extract all the growth parameters involved in the formation and shaping of that leaf. Once you have that, you can then start to come up with models for how the leaf gets shaped and how the cells get divided as the leaf is shaped. And that's illustrated here. So here's a model now in which when we run this model, we'll see that the leaf is growing, the cells are dividing according to certain rules, and the pattern, the shape and the pattern of cells reflects very much what you saw in that movie. All right, so we're now capturing much of the behavior that we see experimentally, but using a growth model based on the principles that I've been telling you about. And the heart of this model is a polarity field, a planar polarity field. That's what's orienting the growth. It's these arrows, which are black in this case, but they correspond to the blue planar arrows that I was showing you earlier. They're orienting the growth that allow, for example, this narrowing to occur at the base of the leaf, because growth perpendicular to these arrows is reduced at this end. And that's why you get a PTO. So this shaping of the leaf is really guided or depends upon this polarity field running through it that's helping orient the growth and allows you to shape it in this way. Now, we proposed this model about 8 years ago, but people said, look, it is all very fine, but there is no evidence for any of these arrows. Where is the biological evidence for these arrows? And in fact, they were right. If you look at a leaf, and here's an Arabidopsis leaf again, if you look at the cells, this is now in a mature leaf on one side, you'll see that there's no obvious polarity, there's no arrow, there's no difference between one end of this leaf cell to the other end of this leaf cell. Okay, or this cell, all right? They all seem to be like rather symmetrical jigsaw pieces that look the same from top to bottom. But then a few years ago, we introduced another fluorescently labeled gene called basal. And when we introduced this gene, which was fluorescently tagged into the leaf, what we saw was that it only labeled one end of the cell. So if you look at this cell, for example, the signal, this orange signal, not orange, it's sort of greenish signal, is only at this bottom end of the cell. It's not at the top end of the cell. This one is the bottom end of this cell. And so you can see, using this marker now, we can assign a set of arrows. Every cell actually does have an arrow. It has one end that's different from the other end. It has a polarity. And moreover, you can get the average polarity across the tissue. You go over the entire leaf and you quantify this polarity. And what you find is you end up with a field of arrows. And this field of arrows is very similar to what we'd predicted by the model. So we now have direct evidence for these arrows, this polarity, exactly as we might have predicted, further sort of confirming the validity of these types of models. Okay, so what I've shown you then is using the principles that I've outlined, we can explain how something like a flat leaf of Arabidopsis, how the sheet is formed, and how the sheet is then shaped to produce a leaf with a particular outline. So we've seen how the principles that I've described can account for the development of something like a flat leaf. But not all leaves are flat. Some form quite complicated shapes, like pitcher leaves. So the question is, do the same principles account for these more complicated shapes? And several years ago, I thought it might be fun to try and investigate this problem using the leaves of carnivorous plants. And very fortunately, the ERC saw the potential in this project and kindly funded it. So what I'm now going to tell you about is really a result of this ERC funded project. Here you can see 4 cup-shaped leaves in carnivorous plants. They've all evolved to trap animals within these cups. Right? And what's remarkable is that this has happened 4 times independently. These have arisen 4 times quite separately from flat-leaved ancestors. All right, so the question is, how are these cup-shaped forms that we see here, how did they evolve? And how is it that it happened so many times independently? And we've been investigating that problem by studying this species, Utricularia, or bladderwort, which is convenient because the cups are much smaller than the other species. They're only a few millimeters long, and they can be imaged quite readily under the microscope, allowing us to see exactly how this cup-shaped form arises. So just to show you what this Utricularia looks like in 3D, here's a 3D movie of the bladderwort that we've taken. You can see it looks quite ominous. It's only, remember, a few millimeters big, so it's not quite as scary as it looks. But if you're a small crustacean and you're unfortunate enough to swim by and trigger one of these hairs, then you'll get sucked inside and get digested, unfortunately. So how does something like this develop? Well, we can look at the cells at an early stage. You see it starts from this small bud and it forms like a cup shape. This is in section. It forms a cup shape, then almost folds over to almost a spherical form. And then finally it enlarges like this. And this little gap here is the mouth that will form the mouth into which the prey gets sucked. So how is it, first of all, that a cup-shaped sheet arises? And that seems to be quite different from the flat sheet that we've seen in species like Arabidopsis. Well, we can turn to the genes involved in sheet formation because we know quite a lot of them, as I said, in Arabidopsis. So just to remind you, we have these genes in the upper surface or the upper region called the adaxial genes, and the ones in the lower surface called the abaxial genes, and then the midplane between the two. So one of the questions we asked was, where are these genes active in something like Utricularia that forms a trap? Where are they active in the primordia that form the trap? And so we tried to localize where they were expressed using various probes, identified the corresponding genes from Utricularia, and the results are shown here. So the signal, this for example is looking at the blue gene, the adaxial genes on what's in the upper surface, upper region of the Arabidopsis leaf, and the signal is shown as this darker color. Okay, and I hope you can see that the blue gene, as it were, is expressed preferentially on the inside of our trap, on the inner surface, whereas the brown gene, the abaxial genes that are normally on the bottom of the flat leaf, are expressed on the outside layers of our trap. Okay, So we can summarize that by this arrangement. So here's a flat leaf where you have the blue on the upper, brown on the lower, whereas in Utricularia, in a trap, we have brown on the outside and blue on the inside. So the blue inside corresponds to the upper surface of the flat leaf. And the question is, how did this arrangement arise based on the principles that I've been describing? So let's go back to our sphere and remember that to generate a flat leaf, we had a polarity field, these black arrows, orthoplanar polarity field emanating from this green midplane. And that was enough to generate a flat sheet. Supposing we were to displace this midplane so that instead of running through this along the equator, it displaces northwards at its edges. So we formed a kind of curved region here, a curved green interface with the adaxial, the blue domain, being in a smaller part compared to the brown lower domain. Well, if we run exactly the same model as before, but now with this displaced midplane, what shape will we get? So let's find out. So this is the model. We're now running it with a smaller blue domain than we had before. If you run that, you generate a cup shape. If I just show you this with a sphere, you'll see the entire cup. So actually, it turns out to be quite straightforward to generate a cup. All you need to do is to have a smaller adaxial domain, a smaller blue domain displaced, and you will generate a cup very simply. And that makes a very strong prediction. It predicts that the blue domain in the Utricularia primordia should be smaller, displaced and smaller compared to the blue domain or adaxial domain in the Arabidopsis leaf. And this is the data. So here's an Arabidopsis leaf. I hope you can see that the brown signal, the stronger signal, the boundary runs pretty much through the leaf, through the middle of the leaf, whereas in Utricularia, it occupies a smaller domain, all right, which is consistent with with the model that I've just given you. So it turns out that it's very straightforward now to generate a cup-shaped leaf from a flat leaf. All you need to do is to displace this adaxial domain, this blue domain. And maybe that's why it's happened so many times independently. It's actually not very difficult to generate a cup. You just need to make this shift in gene expression. And you can only understand that once you understand how these polarity fields are controlling the growth. And in fact, it turns the situation on its head, because it's harder to make a flat leaf than it is to make a cup. Because for a flat leaf, you need to make sure the plane goes through the middle, whereas for a cup, all you need is to be displaced at one end. So actually, in some ways, from a developmental point of view, cups are easier to make than flat sheets. So we've seen how the cup-shaped leaves of carnivorous plants can be generated by the principles that I've been talking about. But these leaves are not simply cups. They have themselves quite complicated shapes. And you can have, of course, cups in many different forms. So how does that shaping happen? And again, we're going to go back to the Utricularia to look at its later stages of development, to try and understand the principles by which the more complicated shape of the cup is formed. As you may recall, after forming a cup, the early Utricularia bud transforms itself into this kind of almost spherical shape, which we're seeing in section. And then from this sphere, there is a further deformation and growth, which leads to the mature trap, all right, with its characteristic mouth for trapping animals. And during this process, some regions grow more than others. For example, this magenta region, which is in the bottom, just below the mouth, like the chin of the trap, grows quite straight and long to form this elongated edge, faster than this red region around it. So you end up with an alteration in shape because some regions are growing faster than others. The question is, how does that happen? And how can we explain what's going on? So let's start off with a simple model to try and get our head around what's happening. in this model, we have a sphere. We're going to approximate the early structure as a sphere, and we're going to divide it into domains, like the magenta domain and the red domain, and see if we can, just by playing with these domains and influencing the growth rates within these domains, we can generate the transformation. Now, although this is a sphere, unlike the spheres that I was showing you earlier, this is not solid. All right, it's got a hole in the center because we're dealing with this cup-shaped form that has arisen, that's turned, as it were, into a sphere. So it's hollow in the center. And we can also decorate the sphere with cells. We can put cells over the sphere so that we can monitor how these cells also get deformed and change shape as our tissue grows. So the first attempt at modeling, we tried to just see if we could explain the shape transformation by playing with how fast different regions grow relative to each other. And what you're going to see here is our sphere growing with different growth rates, particularly higher growth in that magenta region that will form the chin. And I've color-coded the cells according to the area of the cells. Okay, so let's see what happens if you try and simulate the growth that way. So you see the tissue grows, the cells get bigger, but instead of generating a nice straight chin, what we see this bulge forming. All right, we're not forming this nice elongated straight edge. And the reason that bulge is forming is because if you increase the growth rate in a particular region like this, and growth is equal in all directions, it's isotropic, then the tissue will bulge out. All right, it's just going to happen automatically. So how is there any way around that? Well, again, the trick is to play with polarity. So imagine now we have a polarity field in our tissue, just like we had a polarity field in our Arabidopsis sheet. But now the polarity field is over a sphere, over a curved surface. It's the blue arrows, the planar polarity. And let's suppose it's running from the stalk towards the mouth. And now we can have very similar rules before, enhance growth, for example, in this magenta region, but have the additional rule that growth is going to be higher parallel to the blue arrows than perpendicular to the blue arrows. So that's going to orient our growth. Okay, if we do that, then we can have a much straighter edge. We're starting to generate a shape that's much more like the trap. So just by playing with polarity and having this anisotropic growth, we can now generate a much more realistic shape. So is there any evidence that this is correct? Well, one of the predictions of this model is that it predicts that the cells are much more elongated in this chin or ventral domain. And I've color-coded here the cells according to the degree of anisotropy. So the more elongated they are, the yellower they are. So you see, we predict a set of yellow cells in this region, highly anisotropic, but we can actually measure the anisotropy of the actual cells in the utricularia by imaging them. And this is the data now. This is viewing it viewing the ventral side. This is viewing it from the side. And you can see that indeed the cells are elongated in this region, in particular, the region we predicted based on this model. So that's gratifying. We're seeing correlation or a corroboration of our predictions based on cell shape. But what about the polarity? We're postulating there are these hidden arrows, these orientations within the tissue. Is there any evidence that that's the case? Well, in utricularia, if you look in the inside of the trap, what you'll see are these hairs all lining the inside of the trap, the internal cavity. And if you zoom in on those hairs as shown here, you'll see that two of the arms are more splayed out than the other two arms of each of these hairs. And that allows you to assign an arrow to each hair, a polarity. The hairs have a polarity. That's a bit easier to see on the next slide. Here we've got a 3D image of one of these hairs. And you can see that the two of the arms are more splayed out. And then we can place points on these hairs and automatically then extract polarity, an arrow from each of these hairs. And if we then do that for the entire trap, all right, we go through and we place these points and define these arrows on all the internal hairs in the trap, we can then get their field of polarities, the set of orientations as a whole. And if you do that, then what you find is that the arrows have a coordinated pattern, and that pattern tends to be that they point from the base of the flower and the stalk towards the mouth, which is exactly what we predicted based on our model. All right. So that's another prediction of the model that's borne out by the experimental data. So what I've tried to show you today is that armed with these concepts of regional identities, polarity fields, and mechanics, we can explain not only origami forms, but a whole range of biological forms. And moreover, it's allowing us to test these models and make predictions that we can validate with experimental data. So we're starting to build up for the first time a coherent logic and experimentally based set of models that allows to explain the range of different forms that are generated. And of course, that raises a whole set of new questions. What controls these polarities? How are these regional identities connected with these polarities? How are these polarities influencing the growth and the deformation of cells? These are all exciting questions that lie ahead. And so that eventually maybe we'll have a fully mechanistic understanding of how these remarkable and beautiful structures are produced. Everything I've told you today was the result of a team effort, people coming together from different disciplines and working collaboratively to produce the results that I've shown you. So before finishing, I'd like to just thank the people involved. First of all, I'd like to thank Richard Kennaway, who's seen here in between programs having a bit of light relief. He's a brilliant computer scientist, and without his contribution, all the modeling that I've been showing you about growth really wouldn't have been possible. So I'm enormously indebted to Richard. I'd also like to thank many other members of our group. Here we all are having tea together. This is in the pre-COVID days. There's Beatrice Pinto-Gonçalves, Chris Whitewoods, and Karen Lee, who really did the work on the Utricularia project and developed that whole set of work results that I showed you. Kathy Mansfield, who did the work on quantifying the polarity of the basal protein in Arabidopsis, and Sam Fox, who did the work on quantifying the cell divisions and the growth, tracking the growth of the Arabidopsis leaf. I'd also like to thank Geordie Chan, shown here hugging a fellow person in the lab, who was the first really to demonstrate that basal showed this coordinated polarity in the Arabidopsis leaf. And many other past members of the group who contributed in various ways to this project, particularly Jay Cheng, who did the modeling of the 3D cup that I described. Finally, I'd like to thank two collaborators, both computer scientists, Andrew Bangham and Przemysław Kruszynkewicz, and both of them have been inspirational in the discussions and the ideas that we've bandied about. They've been fantastic set of collaborators, and many of the ideas that I presented really arose through discussions with them. So I'm eternally grateful for that. So on that note, I'd like to finish and thank you very much for listening, and I look forward to all your questions.
Okay, thank you for this very interesting seminar, this great presentation that you shared with us. Now we will have some time for a Q&A, okay? So I invite all of you, the audience, to use the chat of YouTube to write your questions. And while we wait for this, for you to write questions, I will start with the first questions. The first question for Enrico, okay? You have described this very interesting mechanism to define shape. The question is, how similar are the developmental processes that you have been describing between plants and animals?
Thank you. Good question. Well, it kind of depends on how the scale at which you look at things. So let's suppose if you zoom into the cells, plant cells and animal cells, and you compare their behaviors. The fundamental difference is that plant cells are fixed. They don't slide relative to each other. Okay, so that means that essentially all the deformations in plants are caused by growth, by some cells growing more in certain directions rather than others. If you zoom into animal cells, you'll see that in addition to growing, they can slide. So they can slide relative to each other. So that means that they can deform in two different ways. They can deform by growing, like plants, or they can deform by sliding and also by contracting. But now if we zoom out, I didn't really talk about cells very much in my models. I zoomed out and talked about the whole tissue. If you talk about the whole tissue, then the difference fundamentally between an animal and a plant tissue is that you'll see that animal tissues can contract as well as grow. For example, there's a thing in animals called, an animal development called convergent extension, in which cells kind of converge in one place and, sorry, you can't see, can you? Cells converge and extend in the other direction. And essentially at the level I'm talking about it, that would correspond to shrinkage in one direction and growth in the other direction. So fundamentally at the scale of the tissue, the only difference between the plants and the animals is that animals can shrink as well as grow. But fundamentally all the other principles are the same. So the two polarities, for example, that I talked about orthoplanar polarity. That often corresponds to what animal biologists would call the apical basal polarity. And the planar polarity that I'm talked about corresponds to, well, it's called planar polarity in animals. So actually, at that level, at this broad level, the two systems are incredibly similar. I would say the same principles are involved, except that you have this contraction as well as growth in the case of the animal.
Okay, thank you, Enrico. Maria.
Yes, so I would like to ask a question that I sort of prepared and thought about. So I'm going to just share my screen with everybody. So hopefully now you can see the photo of a very interesting orchid. And what you can see in this orchid is that one of the petals looks very much like a wasp, in fact, like a female wasp. And in this particular species of orchid, male wasps are attracted to this orchid flower, and they come and they try to mate with this, well, let's say mimicked female wasp, and in that way help to pollinate and propagate the orchid. So my question to you is, you've been telling us about how these complex forms and shapes form. What would you say to somebody who asked you, how could such a shape develop through evolutionary processes rather than by design?
Oh, very interesting question. So I think you're talking here about the evolutionary timescale, not just the developmental timescale. So I talked about development, basically, which operates within a generation. But you're, I think, asking about how do you evolve a particular developmental path, particular way of developing. And the bee or the insect and the plant and the flower, you have to think in evolutionary terms, where it came from. So originally we had, well, in most cases, you have a flower and a pollinator, usually a bee. And there's a mutual benefit between the two, so that the bee gains pollen or nectar by visiting the flower gains by the bee transferring pollen to other flowers. So both are happy, both gain. And that's fine, except that in evolution, so both have costs as well as benefits. So for the flower, the cost is to make the nectar. The benefit cost is the effort of growing in the flower. But in evolution, you'd expect such relationships to be quite fragile, because if one of the partners can get away with the benefits without the cost, then that will be advantageous for that partner. So in your case of the orchid flower, what's happening is the orchid flower is not producing nectar. It's cheating, in a sense, cheating the bee. So the bee is coming, all right, and doing all the work of the pollination. It's getting no reward whatsoever apart from frustration, okay, as it tries to propagate with this flower. So the flower is cheating. That's one way of thinking about it. Now, the other way, the bee can cheat, by the way. There are plants in which the bees will visit. They'll steal the nectar without entering the plant because it's quicker.
Mhm.
The bee cheating. So in evolution, you'd explain these relationships as basically one partner trying to treat the other. Now that's very common in human behavior as well. Suppose you have a very powerful president, for example, and this president gets elected on the basis of mutual benefit. All right, that the people that elect him expect benefit from the president, and the president to be honest with them. All right, and the president gets benefit as well by being in a position of power, but that's subject to cheating. Well, let's get away, finds that they can lie and cheat, get all the benefits without any of the costs. Okay, so it's the same process. So, but that's happening on a human timescale, whereas I'm talking about it on an evolutionary timescale. So, from an evolutionary point of view, it makes perfect sense that you would end up with this cheating. Actually, it's harder to explain on design. If you think why, let's say, if you had a sort of a creator creating, why would it create cheats? Why would the creator be a nasty creator? Why not have every animal and plant interacting beneficially? Why create this cheating relationship? Well, actually, it makes much more sense from an evolutionary point of view, where you'd expect cheating to evolve compared to a design point of view, unless you're a rather nasty designer that likes to put cheating into your system.
Thank you very much for that question. So I can see that we have some questions now from the audience. So Alessandra from the ERC, she's asking, it would be interesting to know more about the parallels with human physiology. Do folding errors always end up in miscarriage, or do we see malformations that can still result in live births?
Oh, that's a very good question. Well, I don't know if you remember there's a famous quote from a biologist, Lewis Wolper, who said, the most important event in your life is not birth and marriage or something like that, but gastrulation. And gastrulation is a very early folding that happens in animal development. And if that goes wrong, basically, you'll get an abortion. The thing will not grow. So yes, folding errors are absolutely critical. If a folding error happens, then the embryo will not develop properly. So yes. So these events are often going to be preterm events. So that these abortions will usually happen, or as you'd say, arrests of development, you might say, will happen. Even within the first few months. So the pregnancy will terminate. Because they're so fundamental. If you start folding things in the wrong way, you basically can't go very much further. So I would say, yes, they're fundamental. And the principles that I've talked about, as I said earlier, are very much the same as in humans. I would expect them to be. That's a hypothesis. Are going to be very much the same in humans as in plants, except for the difference between sliding and non-sliding. So yes, I think they would be critical for. We need to be folded in the right way to be here. We would not be here having this conversation if we'd had a folding error in our early embryogenesis.
So another question that we have is: are there any systems in which allow polarity suppressing in two different directions, and nevertheless give rise to the same shapes?
Now, I'm not entirely sure what the questioner means. I guess, according to the principles that I've described, if you had polarity broken in both directions, you'd stay as a sort of blob, okay, because you wouldn't be able to orient your growth. If the question is asking, is there experimental evidence? What is experimental evidence for something that gets rid of both polarities? We don't have mutants in plants, at least that I know of, that give you just blobs, or rather, we have mutants that give blobs, but whether they are blobs because they don't have both polarities has not been established.
So that question came actually from Wieland, who I know is a physicist, and he has a sort of a follow-up, and he says, a number of figures and geometric operations you explained remind me of symmetry breaking in physics. How do you characterize the symmetry breaking in the field of research? Okay,
I think that relates to how you establish let's take the simple example I gave at the beginning, where we had a mid-plane organizer. Okay, and we ask about the symmetry. Well, the first symmetry I'm breaking is putting a mid-plane organizer in the middle. All right. I start with a sphere, I put my midplane, and then I can get my polarity, but the polarity is oriented in relation to the midplane. So it comes to the question, how do you establish the midplane? And there are a number of models or mechanisms in both plants and animals that discuss how you break symmetries and establish different regions of activity, such as the midplane region, which often use cell signaling and patterning. So that's what I would call patterning mechanisms and domains of gene activity, which that's a whole topic in itself. But there's a lot known about that. There's a second question, which is how do you go from a pattern of regions into morphogenesis, that is to orient your deformations. And that's what I was talking about. So I kind of started with an assumption of a pattern, which in this case was the midplane, and went from there to say, how do you go from the pattern into the morphogenesis? But I'd already broken my symmetry by having my regional identity in the midplane. So I don't know if that answers.
Okay, now we have another question from Jaime. He asks, how could you see this research being applied? And he also says, thanks for the very fascinating presentation.
How could you see it applied? Well, in the broadest sense, it relates to that. It's so fundamental what I'm talking about in terms of the principles. It relates to animal development and plant development. And it relates to that question, for example, about the human embryos. So in that sense, it gives us understanding of how development occurs. And it can be important if you want to then engineer development, or for example, if you wanted to have regenerating systems, regeneration of tissues and whatnot. But I would say the applications are kind of like at this stage are very difficult to fully appreciate. It's a bit like if you think Michael Faraday, he discovered electricity, and the applications of that fundamental discovery took many, many years. And now we couldn't live without electricity. So in a similar way, we're trying to understand the fundamental principles, but how that will ramify into the way we think about plants and animals and interact with them, I would be very hard pressed to predict, other than from understanding. And in fact, actually, I believe this is the ERC philosophy, that we really need to understand, and by understanding, then we can apply. But those applications, in a sense, are just as difficult to anticipate as, in some ways, as the understanding in its own right.
Okay, thank you. We have another question from Tanguy. Could there be some kind of learning from the mathematical discipline that's called topology? Could this be somehow related to what you're talking about?
Oh, it's an interesting thing to think about. I'm not an expert in topology, but clearly there are topographic changes going on as the system develops. So I don't know is my simple answer. I think in principle, you might expect there to be some relationships, because topology is all about conformations and connectivities. I should say maybe there is an area where topology has been massively employed, and that is in relation to cell divisions. It's not something I talked about. So I talked about the deformation of sheets. And that, as I said, the topology implications, I don't know. But when you look at cell divisions, then you also have a topological problem, because as cells divide, they create new vertices, and they have topological relationships between cells and neighbors. And that aspect, which I didn't touch upon, although you did see some models with cells growing, there have been sort of investigations of topology in relation to cell connectivity.
So I've actually just received a question via SMS that came from Mariam, who you may remember because she was the scientific officer working with you for LS3 panel. So she wants to know why you chose Urticularia as your model system.
Interesting. Well, We did a lot of work on flowers. Now, flowers have as we've just discussed, a mutualistic relationship with insects. In fact, if you look at plants, the most complicated structures in plants are to do with animals, dealing with animals. A flower deals with the pollinators and is basically, and you could see it from the flower's point of view, the flower is manipulating the pollinator to do what it wants using this complicated shape. Now, the other classic case where plants manipulate animals through their shape is carnivorous plants, okay? But in that case, it's a one-way relationship. There's no benefit usually to the animal because the animal gets swallowed up, but there is a benefit to the plant, okay? So that's why you find these other complicated shapes, and they've all evolved from leaves. So I was interested in how these shapes evolved from leaves. And I was thinking, well, the trouble is that the 4 examples that I gave you, 3 of them are very large cups, but only one of them had these small cups that looks like it might be worth pursuing in terms of imaging. So I thought, well, that might be a tractable system, although there was no genetics at that time in that system. So then I thought maybe this could be something we should look at. And I thought, well, you know, if I was thinking, in fact, of putting forward an ERC proposal, I think, what about ERC? Because this is way out there, you know, this is not something. And I thought, oh no, the referees are going to say, what's the genome size? They're going to say, okay, you can't work on this because it's got a massive genome. There's no future in it. So then I remembered it was Sunday, okay, and I Googled, I'll Google it. I'll Google the genome size of Utricularia. And came back, smallest genome of any plant is Utricularia. And it was like, whoa, sudden shock. And it was on a Sunday, all right? So if you're religiously minded, which I'm not, at this point, you might think this is an act of God, that suddenly Utricularia turns out actually to be the smallest genome ever, at least ever measured in plants. So at that point, I said, well, now I have to do it. Okay, so a sign has been sent that this is the smallest genome, so I should do this. And so that's when I decided I would definitely put the proposal forward and work on Utricularia.
Mm-hmm. So we have another question from Federico that says, he was wondering, can fractal patterns that we observe in nature, for example, cabbage, be explained by the mechanisms that you were illustrating?
Yeah, and again, these are all great questions, by the way. So fractals in plants have been traditionally studied very intensely through these branching algorithms. So it's to do with self-similarity. That is, so a branch, if you think of a branching tree, for example, each branch gives rise to another branch, which gives rise to another branch, and they can be self-similar in terms of different levels of branching. And there have been systems called L systems that have been developed, particularly by one of the collaborators I mentioned, Przemysław Prusinkiewicz, who's done a lot of work on these fractal patterns, these self-similarity patterns that arise through reiteratively using the same branching rules to generate these fractal patterns. So that's why, in fact, if you look at plants, fractals are everywhere, but they're most striking in terms of the branching, I would say. So yes, so although actually you could say that even something like a growing cabbage or something like that, which has a wavy leaf, even there you might imagine that you could think of it in fractal terms, because what you're doing is you're essentially applying the same growth rules again and again and again as the system grows, and so you're generating these patterns. So in that sense, in a very broad sense, that's self-similarity as well. But for sure, the fractals as we know them are very closely related to branching patterns that we see in plants.
So we have another question from Michelle. Is there some randomness in growth?
Yes, is there some randomness in growth? I would say absolutely yes, but it depends what you mean by randomness. So there's certainly statistical fluctuations. Every process, every biological process has a noise. There's a noise in any system, okay? Now, we can call noise randomness. So that, for example, the growth rate of one cell is not going to be exactly the same as the growth rate of a cell that's next to it. So there will be fluctuations. There's another type of randomness, for example, and for sure that is happening as things grow. Okay, so what we're seeing is a kind of average It's a bit like saying when you take the temperature, all right, you have a fixed temperature, but actually there's a lot of randomness going on because the molecules that are jumping around, and actually, in a sense, their kinetic energy is transferred into an average property, which is the temperature. So all the growth that I'm describing is an average of some randomness. Now, there's another sense of randomness. For example, some leaves, the left half of the leaf has a different size to the right half of the leaf. And in some cases, that can be almost random. It can fluctuate, all right? In other cases, you see it's consistent. It's always one half of the leaf versus the other half of the leaf. So that's another type of randomness. You know, to what extent is something consistent, or to what extent at that scale does it fluctuate from one to the other? Like spirals. Okay, so if you look at a plant growing, you have leaves are generated in spirals. So in some cases, 50% of the plants will spiral one way, 50% will spiral another way. But if you look at something like a bean, a runner bean, it always goes up. I think it's a right-handed helix. I may be wrong. It's either right-handed or left-handed. Always one way, just like DNA. Okay, so again, you can have randomness at different scales. Okay, so right from the molecular scale through the cellular scale and through to the more developmental scale. So randomness for sure is absolutely part of. You can only think in terms of average, or rather, at any scale that you're looking, you will find randomness beneath it.
Okay, maybe I could just make a short comment to that, which is in the nematode worm in C. elegans, there's a very constant developmental pattern. And in fact, people sat under the microscope for many years just watching these cell divisions, and they know exactly how many cells there are in the worm. And there's also this programmed cell death, that certain cells are born and then are killed. So is there anything like that in plant development, programmed cell death?
There is programmed cell death, but it's not as regimented as in the worm. Okay, so in the worm, you've got these very defined lineages. Okay, and then the programmed cell death comes at certain places in those defined lineages. There isn't anything like that because there aren't such clearly defined lineages. But you can have cell death. So the most obvious example is the death that is happening around us now. You look out of your window, if you've got a window, you'll see leaves falling from the trees, and that is programmed cell death. All right, it's not necessarily the same mechanism as in worms, but imagine you were a biologist and you're looking at these things called trees, and they were growing, and you say, oh look, at a certain point in development, all these cells die and they fall off, and then they regrow the next cell. So that's one form of cell death. Another form of cell death is cheese plants or monstera plants. If you've got, these house plants that look like cheese plants, I don't know what you call them. They have holes in them. The leaves have holes in them or grooves in them. That is also caused by cell death. But by and large, if I'm looking at a continuous leaf, that there is very little cell death involved in that. Okay, so there isn't programmed cell death in the sense of forming a leaf, but if you have holes in the leaf or if the leaf falls off, then you're often dealing with cell death.
So I actually have another question also that just came by SMS from Yanni. So he asks how you make a maple leaf, and I'm wondering if he's thinking, well, I'm just imagining now, because I think there's also programmed cell death when you make something like a digits in a hand in animal development. So he asks, how do you make a maple leaf?
Good. So the maple leaf is not due to the death between the fingers. All right. Okay. Although palm leaves, you can have death between the fingers. The maple leaf, actually, even the death between the fingers. Okay. Even in humans, we start off with a continuous limb bud. All right. But there are already axes of growth, and maybe axes of growth in that limb bud. It's not a completely smooth edge. So the maple leaf, we don't know how maple leaf grows, but we know a lot about how serrations and pointy things grow at the edges of leaves. So the most likely way that maple leaf grows is that planar polarity that I described. So if you remember, it was pointing from the base of the leaf towards the tip of the leaf. All right, and that was giving us this. If now we have other points to which the polarity locates, like instead of pointing just to the tip, it goes to two other points on either side of the tip, then you'll end up with like a field of polarities pointing to five, sort of being sucked in at 5 positions. And if you grow a leaf according to that rule, then you grow parallel to the polarities, you'll generate something like a maple leaf. So the maple leaf can be explained by having, instead of one place where the polarity points to, 5 places that polarity points to.
Mm-hmm. So we have a follow-up question from Michel, who was asking about random growth, or whether random growth is possible. And now he asks, why growth at all?
Why growth at all? Do you mean why do things grow?
I suppose it's hard to decipher what exactly he means.
Okay, well, let me answer the very broad way. In evolution, you could ask why evolution, because if you don't, let's suppose you only divided and you never grew, okay, well, you get smaller and smaller and smaller and smaller, all right? So if you take an orange and I cut it in half and I keep cutting, unless I grow my orange, I'm gonna get smaller and smaller fragments. And so if you want to replicate and reproduce, you have to grow, because otherwise you'll get smaller and smaller. Actually, that happens in diatoms. That can happen in diatoms. They get smaller and smaller as they divide. So, so growth is fundamental to evolution. Without growth, you do not evolve, or rather, it's part and parcel of the same thing. Then there's a question why some things grow bigger than other things and so forth. So I can't really. That's in the broader sense of why you absolutely have to grow. But I don't know if there's a more specific question you have.
Okay, so we have another question from Tanguy, and he asked, are there any artificial intelligence algorithms or models which attempt to predict the form of something?
I'm not aware of any, but I wouldn't be. It depends what you mean by predict. Okay, so take an artificial intelligence program. It may be able to predict, but it may not give you understanding. Okay, so and prediction and understanding are not one and the same, although they do tend to go, they kind of get conflated. For example, you can train a network, for example, to recognize the letter A. All right, or you could train a network to recognize maybe the shape of a flower. I don't know currently what the recognition algorithms are used to recognize flowers, but do they tell you how the flower is formed? I don't think the answer is no. They tell you how to classify. They say, I can distinguish this flower from this flower, but they don't tell you anything about how the flower is formed. So they give you a different type of understanding. They're very good at classification. As far as I know, they're not so good at giving you understanding, because understanding, we're doing something very strange. What we're doing is taking something quite complicated and trying to extract some simple principle behind it. We're not trying to just say, here's the world and a very, very complicated description. We're trying to say, we're going to ignore a heck of a lot of stuff and just ask about one thing. If you take the basic one, like gravity, okay? We take it's like an apple and an orange, we drop it, we see they follow the same rules. But actually, whether it's an apple, an orange, a banana, or a football, makes no difference, all right? We're ignoring everything other than the fact that it has a weight and it falls. So actually, you might say, what a ridiculous thing to do. You're ignoring everything except 2 things, just the weight of the thing and the height at which you're holding it. And that's it. So artificial algorithms in general will not do that. They won't extract and simplify. They might say, I can recognize my apple or I can recognize my orange, but they will not arrive at the theory of gravity. So that's the fundamental point, that they will make predictions in a sense. They'll say, I predict this is a banana, but they won't extract and abstract and give you a sense of what we would mean by a scientific understanding.
So actually, maybe I could ask a question related to that. So in many of the proposals that we receive, I noticed recently, especially, at least in my LS2 panel on genetics and systems biology and omics and these things, that there is often a component on machine learning. I have the feeling that maybe it's become rather fashionable right now. Do you have any comment on that?
Oh, I think for sure it's fashionable. Science has its fashions, and there's nothing wrong with that. I think, like with all fashions, people get very excited about it and think that you've got the solution to all sorts of things. I think, to my mind, I'm not an expert in machine learning. I do not wish to in any way disparage it. But maybe sometimes the promise, I think people that do machine learning understand its limitations, what it can deliver and what it can't deliver. People that don't understand machine learning, probably they're going to be more susceptible to thinking, not understanding its limitations or its strengths even. And so as with any tool, you need to know its strengths and weaknesses. And the danger, I guess, from your point of view, is people just throw it in without any understanding. In other words, they throw it in because it sounds cool, rather than saying, which is my case, in my view, incorrect. You should use it if you really understand what you're doing, why you're doing it, what it can deliver, and what it cannot deliver. Then that's an absolutely fine thing to do.
Okay, thank you. I'm not going to take the question from Tanguy that mentions apoptosis, because I think you covered that when we talked about programmed cell death. It's just another word for it. We have a question from Anna Elizabeth saying, do you think the growing vascular system plays a role in the establishment of polarity in the leaf, or is its pattern a result of polarity?
Well, therein lies a long debate. So some people are convinced that the vascular system drives everything. And other people are convinced that it was the epidermis that drives everything. And then there's this rather fuzzy, wishy-washy thing that says they maybe talk to each other and there isn't a single driver. So it's easy to take the wishy-washy position. And I think I'm probably more towards the vascular following the tissue rather than the other way around. Okay, so I would say it's a bit of both. But in terms of my position, I would move probably to say that the vascular is plugged. So the tissue, well, we know that a primordium grows before the vasculature appears, but you have to connect the primordium vasculature to it's like you've got to plug into the previous vascular system. So I would say the two are totally interconnected, but probably the form of at least the earlier stage, the vascular gets plugged in to the growing tissue rather than the tissue being driven by the vasculature. That's the way I would think about it. But I don't think it's completely resolved. I think it's still good to have that debate. But I don't think an extreme view of the vascular driving everything, I don't think that works. But I think something in between might.
Okay, thank you. So I think we've come to the end of the questions. I just wanted to mention it's clear that you love beautiful images and forms, and maybe I saw on your website, and now I saw you also had in your acknowledgement section some portraits that you've painted. Mm-hmm. So I would just like to share with the audience, if you would like to have a look at the wonderful portraits that Enrico has made of many well-known scientists, you can go to his website and see that he's also a very talented artist. Can I give the floor to Jose? Jose, you're muted.
Thank you, Maria. Thank you, Enrico, for this very, very interesting seminar. We just come to the close of this seminar, and let me just first thank Maria for preparing the seminar and also for moderating the questions, as well as the people that are behind the scenes, Oliviero Ferrareto and Paolo Borghesi, that they have been supervising our YouTube ERC channel, making sure that everything is running well. Thank you for your help. And again, I mean, this I think we all enjoy this excellent seminar that you have prepared. We really enjoy the figures, the choreography, okay, that you prepared for all of us. So thank you for coming to ERC to share your knowledge with us, and we hope to see you again in the future, okay? And for all the others, remember that we will have soon another seminar, okay? So stay tuned and join us in the next one. Thank you.
Thank you.